Okay, now we are live.
Okay.
Okay. Welcome everyone to our platform, Bioengine. This is our 24th webinar, and we are very fortunate that Dr. Ricardo accept our invitation. And today, in this platform, he will present his work. He's very renowned, and he's working so dynamically, as I know him last 2 and more years, and I follow him here and his work on the rise and specifically for biotic stress tolerance rise. Today we have our team, Jacinta, Liberty, and Soma. I request Liberty to introduce Bioengine and our initiative. Then Jacinta, please introduce Ricardo. Soma will interview Ricardo after his talk. We collect all the questions from the YouTube chat box. You not need to paste multiple times of the same questions. It's 2 times more than enough. We will collect it and sort it out, because every question is not possible to answer in that specific time. But we'll send all the questions to the speaker. Liberty.
Thank you, Shobha. Welcome to the Bioengine webinar series. The webinar series has been designed to build a platform for where plant scientists can present their research to the world and future scientists can gain perspective and inspiration. The appreciations that we receive from our global viewers after every webinar keep us motivated. We have started a new series called the Bioengine Live Discussions. This is mostly a peer-to-peer discussion section dedicated to research in the field of life sciences. This new series is also a part of the Bioengine initiative. The discussions will include important aspects of our research life, like research planning, conceptualizing, methodology designing, manuscript writing, finding appropriate journals for publication, scientific communication, national and international fellowships for PhD and postdoc research. You are welcome to join this initiative and participate in these discussions on our YouTube channel, and also post your ideas on the topics you want to discuss in live sessions. The next live discussion is on the 12th of December, 2020. Please visit our website to know more and to register. Now, coming back to today's event. Today is our 24th webinar of the series. We welcome all the viewers who have joined us from around the world. After attending today's talk, you can apply for a certificate of participation via the feedback link and password. We have already emailed the feedback certificate application link to the registered participants. The password will be provided multiple times in the YouTube chat box during the Q&A and interview session, which occurs after the lecture. Submission of the feedback form is mandatory to receive a certificate. When you fill out the feedback form, please mention the full name of your institute and the full institute address. You can collect your participation certificates after 2 days from our website. Bioengine does not send certificates through email. Please visit our website to register for upcoming webinars. We can connect with Bioengine via Facebook, YouTube, Twitter, and LinkedIn. I now request Jacinta to please introduce our speaker for today's webinar.
Thanks, Liberty. The topic for today's webinar is From Breeding to Genome Editing, Long-Term Strategies to Contain Plant Diseases by Ricardo Oliva. Dr. Ricardo Oliva is a senior scientist at the International Rice Research Institute, or IRRI, in Los Banos, the Philippines. He is the cluster leader responsible for 30+ staff, including scientists, postdocs, national staff, and scholars. He has over 10 years of research and development experience in staple crops in Asia, Africa, and Latin America. He is the recipient of research funding from key donors such as the Bill and Melinda Gates Foundation, the Biotechnology and Biological Sciences Research Council, BBSRC, and the United States Agency for International Development, USAID, among many others. He has coordinated scientific networks, including public and private enterprises. He has a strong publication record, developed online tools, as well as several patent applications. He has published in journals including Nature Biotechnology, PNAS, Frontiers in Plant Sciences, The Plant Cell, Molecular Plant Microbe Interactions, Crop Research, Frontiers in Microbiology, the Plant Pathology, the Plant Journal, just to name a few. He has also published many book chapters and other publications. He has always been engaged in collaborative research with renowned research institutes from all around the world. We are honoured to have him with us today. I now request Dr. Oliva to please share your slides and you can begin your presentation. Thank you.
Yes.
Okay, sorry. Okay, you hear me?
Yes, yes, loud and clear.
Thank you. I want to thank all the people that is now in the broadcast for attending this presentation, and of course the group. You guys are doing a fantastic job. You know, it's like a very nice way to democratize science, and that's have a big value for all of us, and also in this moment of our lives. Yeah, thank you. You're awesome. Anyway, so today I wanted to talk about the strategies to contain diseases. And when I say strategies, what we intend is durability, long-term, and efficiency. Because if you notice, diseases are still there, and we have been through many processes of trying to develop these strategies for the last 60 years, and we still have the same diseases, just hitting a little bit worse than before. So there's something that we are doing wrong, definitely. Okay, so now it's blocked, sorry. Okay, so there's common diseases that are in the news normally. You can see this banana disease in Colombia, the Fusarium spirorum reaching Colombia for the first time, just setting a lot of alarms in the region because of the production. No, sorry, I have to. Looks like it's blocked. Okay, I have to go this way. In Bangladesh as well, wheat blast just hit in 2017 and set the alarms. In Europe, we have a lot of farmers in the olive industry are really suffering. And these are outbreaks that hit a global perspective. However, the outbreaks that we are looking for are the small ones, the ones that are building up on something bigger. And those are the ones that hit most of the farmers. Imagine at IRRI, what we are doing is trying to work with smallholder farmers. Around 164 million of farmers are out there in Asia. So if you are in that group or in that category, it's highly likely that you have or are close to the poverty line in most of those cases. So any event of crop failure or peak in the food crisis or in the food supply really affect you and affect the household. So those are the group of people that we are intent to help, and those people suffer small effects on the diseases at low scale. Just to give you an idea on the economic impact, this is a publication that was out in 2019, and this is a survey of a big survey on different crops, 5 main crops in different regions of the world. And if you have see here in the world, they measure the yield loss due to pests and diseases. And rice is actually the most affected. They calculated 30% of yield loss in rice. This is a cumulative, so that means that all the pests and all the diseases that are out there every year are taking 30% of the total production. So you can imagine that amount of income and amount of resources. The second point is also the environmental cost. For the environment, the farmers and the environment suffer a lot. You can see here a typical picture of farmers spraying chemicals. And here you see a comet test from a human cell that has been exposed to nothing, and a human cell that has been exposed to a chemical that is common in the agriculture. So that just measured the DNA damage. So definitely there is a component that human cost there. And if we want to understand the pathogen, we can just classify these into two main features. And those are the features that we need to attack or try to make solutions. So the first problem is that pathogen change. It just matter, is a fact, biological fact, right? Pathogens have the smaller genomes, are smaller, and can replicate. The life cycle is small. So they can accumulate mutation, and then they can evolve faster than the host, for instance, the plant. So if you have a mutation in a pathogen and make this pathogen virulent, this will accumulate during the seasons until the outbreak comes. This is the point in we see the disease, we go there, collect samples, try to work what is the relevance for those strains, try to understand what resistant gene we can use, and then we come back to the farmer after several years probably. But by then, the pathogen have changed already, so the cycle is never broken. So what we want actually is to try to detect these virulent forms very early in the process, right? So to do that, we need a lot of information on the genomics. We need a lot of information on the genetic component, the genes that are doing a part of the virulence. And based on that, what we need is to apply a small test or very reduced effort to detect those in the field. Try to make interventions early on in the process to break the cycle as fast as possible. So if we can detect the pathogen early, we can act early and break the cycle. So that's the first thing. The second problem is that pathogens are not static, right? Pathogens do not have passport. They can freely roam around across the regions. However, the countries, the breeding programs, the National Extension Service, they do have passports, they do have borders. So any intervention that they take is local intervention. And you can see here in this test, in this map, Thailand, Cambodia, and Vietnam all have the same pathogen with a very similar virulent spectrum, right? You can see the photos here in Cambodia or in Vietnam. However, the interventions of these three countries are different. So definitely we need tools that help us visualize the problem and take decisions together, right? So that's the key point, to make interventions that have a regional context. Okay, before moving forward, I want to touch a little bit about cellular level. Why we say this is a co-evolving process between the plant and the microbe. And there's a lot of literature out there, so tons of papers. But this is what we know so far. So this is a very simplified way. Sorry if I have too simplified, but what we know so far is that either bacteria, fungi, or oomycete, or even nematodes approach the plants using effectors. The effectors are proteins that are secreted. In the first phase, what they are doing is simply help the pathogen to reach the cell, right? But in the first level of defense, the plants evolve these membrane receptors that the main point is to detect effectors and trigger a defense response in the plant. We call PTI. This defense response is the reason why all the plants are green. It will take out all 99.999% of the pathogens and keep the plant safe. Okay? So, but there is some pathogens that adapt to this process, and they develop or evolve effectors that can go inside the plant cell through the type 3 secretion system in bacteria, or the haustorium is a structure that the fungus or all mycet use to invade the plant. So these effectors are basically plant proteins now, and the main target is to block PTI and allow the infection. Of course, they deregulate many of the processes, and that's kind of a subject of big research at the moment. In the third level of evolution, plant evolved the resistant genes that we call. There are NBSLRR type of receptors that detect the effector genes in the plant or the effector proteins and trigger a second type of resistance that we call effector trigger immunity. And this is ends up in cellular death. So basically the plant is a suicide mechanism, so the plant can stop the infection. And of course, pathogens evolve and kind of try to overcome the plant by modulating these effectors, meaning mutating the effector, changing the effector structure, or even deleting the effector. So this process is going on and on in all the host-plant interaction system that we know so far. There's some variations, and definitely there's many groups studying those variations, but this is the general concept. Okay, however, apart from immunity, we have another mechanism that is being studied recently that we call, or I call, starvation, and is basically the plant generating mutations that block access to nutrients. Okay, the main purpose is that the pathogen, either fungal, bacteria, or nematode, got no access to the nutrients, sugar transporters, for instance, or sugar sources. Here is a little bit more of detail of this process, in which, if this is the apoplast and this is the cytosol, I don't know if you see my pointer. You see now, I guess. So infections of bacteria in the plant, what they are doing is inducing sucrose in particular to be exported to the apoplast part for feeding purposes. In case of fungal, what they're doing is inducing glucose or fructose inside through transporters, of course, to feed the haustorium structure here. So this process is very, very common and is being studied right now. And the reason why we are interested in this is because if we try to block this process of nutrient acquisition, it's highly likely that the pathogen cannot adapt to another source of sugars, right? So unless, or in comparison with effector genes or in immunity, pathogen is very good adapting to immunity processes of the plant. However, if you remove the source of nutrients, pathogen will have a lot of trouble to adapting to a lack of sugars, or try to utilize a different sugar. So that process probably is more long-lasting than the immunity per se. So in general terms, what we want is combine these two processes, either immunity and starvation process, or starvation mechanism on the plant. Okay. So what you see now is somehow our equation, a working strategy, in which we have a toolbox of resistance, so genes that we clone, that we create, do genome editing, or genes that we introgress from wild material. You have pathogen surveillance or pathogen understanding, pathogen monitoring. So what are the populations? What sort of races do we have? What sort of virulence factor do we have? And of course, the environment is important. So whenever we want to deploy any gene, we need to take in account the pathogen population and the environment. And every single season or every single breeding season is an opportunity to take a decision, right? So we are applying this sort of decision making to guide our breeding programs, at least at IRRI, and we try to do this with other partners as well. So try to make this like a rule of decision making every season, every breeding block. I will explain how it works and how we want this to work. And let's start from the resistant part. Okay, today I will talk about not breeding per se, but more on genome editing part, which for us is a new breeding technology. I will focus first on bacterial blight. Is a disease that we have been studying for a long time now. It's caused by Xanthomonas oryzae, Pathovar oryzae. It's a bacteria, gram-positive bacteria. And Xanthomonas is a vascular pathogen, so get in the plant through hydatodes or small wounds, and it moves through the vascular system. And what it does basically block the circulation of the leaf, and the leaf die. And this lack of photosynthetic tissue generate yield loss. So far, we have been controlling these bacteria using XA genes, so our normal genes that we put in rice. But what gene goes where and for how long, and what is the durability of that gene, that's a matter of discussion, and it's something that we are working on a lot, or to develop new genes that we know are more durable. So to give you an explanation what's happening at cellular level, imagine that this is the cell and this is the bacteria. So bacteria carry these effectors that are called TAL. And TAL are transcription activator-like proteins. So these are big proteins, actually. Each genome have around 18 to 22 TAL effectors. As a transcription factor, they have a nuclear localized signal and activation domain, but also a DNA binding domain. So this DNA binding domain is built on repeats. Each of the repeats, as you see here, recognize one nucleotide in the promoter region of a gene, right? So it's a very gene-specific or sequence-specific activation. So what happened is that the TAL effector is being secreted inside the cell, go to the nucleus, recognize this site or this sequence, and activate the gene. Now we know that this TAL effector activate genes that are called sweets. These are sugar transporters, and basically what they are doing is export sugar outside to the apoplast. So as I mentioned a couple of slides before, this sugar is utilized by the bacteria. There is a lot of replication, and the bacteria continue with the infection. So it's very, very smart way to approach the cell, and it's subject of a lot of research because of the delicacy and the specificity of this process. Okay, this has been shown several times. This is one of the papers we have been collaborating with. This is lines that have a reporter gene, gas reporting in sweet 11, for instance. And you can see the induction of this gene in the vascular tissue. Probably the cut here is better than you can see. The cells start to induce the sweet 11 when are inoculated with the bacteria. Here you can see the same, but in a time course experiment, 1 day to 5 days post inoculation. You can see here in this part, some cells start to induce the gene at 3 days, but at 5 days is already highly induced. So the bacteria have enough sugar to continue the infection. We also know that some individuals are able to activate particular sweet genes. For instance, this strain that is called PXO71 is able to induce sweet 11, as we see here in these transgenic lines, but not sweet 13 or sweet 14. However, this strain that is called PXO86 can induce sweet 14, however, is not able to induce sweet 13 and sweet 11, and so on. So we know that these three genes are important, and we also know that not all the bacteria have all the genes. So the repertoire of TAL effectors are distributed in the population. But together, like a conglomerate, they are able to induce sweet 11, sweet 13, and sweet 14. And this topic is, again, a very big topic of research about the sweet genes in general and how they facilitate the infection, not only in rice, but in other crops. And you will see some papers going out there on the function of sweets into how they facilitate the infection process. So our working theory, again, we have 5 major TAL effectors from the bacteria side, and we have 3 sweet genes. So what we want to do is to block or to change the EB site in a sense that we block the TAL effector binding, and this binding will block the expression of sweets in the plant, either 11, 13, or 14, or all together at the same time. Remember one thing, that what we want is just touch or block the recognition site, but not messing up with the promoter region. Because if we touch the promoter, then the gene cannot be induced normally, and then there is a trade-off for the plant. So we need to be very careful about that. So our hypothesis is that if we block this process, we may find a way to block the whole sugar or sucrose transportation towards the pathogen infection. So this is what we did, actually. This is a slide, very nice slide from Bin Yang. It's one of our partners in the project, in the work. Just represent what is happening with TAL effector. So TAL-C, for instance, this effector that is in African populations, recognize just 20 amino acid, 20 nucleotide in the DNA of sweet 14. Have a specific target sequence, and we design guide RNAs in the construct that target this region. These guide RNAs, of course, was recognized by the Cas9. The Cas9 is a nuclease that cut the sequence. This sequence is repaired by a system that is not specific in the plant, at least in higher plants. And this non-specific reparation will introduce a set of mistakes. And this is the base of our strategy, that the fact that by chance there are more mistakes than correct reparation give us a lot of room to play with this tool. Of course, one beauty of this approach is that the tDNA insertion happen in one of the chromosomes, and it's highly likely that the target region is in a different chromosome. So you can easily segregate these two plants and continue the T1 with a tDNA-free plant. In here, what you observe here is some of the results for a couple of the regions. What we did is actually target 5 different regions at the same time. I'm just showing 2. In yellow, you see the wild type sequence, the original sequence, and in red, if you can see the red here, you have several results or sequence that we obtained by CRISPR in that region. So for instance, 1A in introduction, couple of T's, 1T. Here in the different sequence, we have 2A's insertion. We have a deletion of 7 base pairs in here, and so on. So we hope that accumulate the most number, or a good number of mutations and decide what mutation can give us what we need. And what we did is that enough difference in the sequence to block the binding, but what we don't want is big deletions that may affect the promoter or may affect some regulatory element that is close by. So we need to be very careful about this. And this is what we did. This is a summary here of the 3 genes and the 5 locations using different regions. And we select these plants on T2 or T3. We inoculate with different strains of XOO. These strains are carrying one single TAL effector for each suite. So what happened here is that we clip inoculate. So inoculate in the top here, and the bacteria move down. And as you see, the bacteria is taking over the tissue. However, in the plant that has been edited, the bacteria is inoculated in the top, start to the infection, probably is very good at the beginning, but soon after the sugar run out and the bacteria die or starve to death, basically, and the infection stops. So you can see the same here in the greenhouse experiment this time, where the infection is almost none in the genome edited lines compared to the wild type. Here are the plants in the greenhouse. You can see they're very nice and happy plants. However, not everything is good. As you know, we are using genome editing. We are using transgenesis. So some plants, we expect that there are a trade-off, different targets or targets that you are not expecting. And also, the transformation itself can introduce some mutations or some changes in the structure of the genome that can change a little bit the plant. And we detect that when we did the agronomic trait of these plants. What we measure is different agronomic characteristic of the set of plants to see if they have a trade-off effect with the wild type. Here's the wild type, and in most of the cases we have similar measurements for yield, fertility, plant height, and so on. We only find one plant out of 20 that shows a little bit less fertility than the rest. And this plant in general is a little bit different. You can see here the panicle is a little bit different, smaller. And this plant, sorry, is this one actually. And you can see that immediately kind of glow as a different plant. At the beginning, we thought that was a contamination, a different plant that we planted there, but no, after the analysis, we realized that this same plant, good mutations because it's resistant, but somehow we hit some part of the process and some clonal variation during the transformation changed the plant a little bit. But yeah, if you want to develop products, you need to go to multi-location trials to understand what is the effect of the field, what is the effect of the environment in the plant, because you don't know what happens when the plant gets stressed by drought, by salinity, or other stress agents, how that plant will react. So what we are doing at the moment, we're working with our colleagues in Colombia, in Burkina Faso, or in the Philippines. Also, we start working in Uganda and India to try to test those plants in a multi-location trial to assess exactly that. What is the effect of the mutations in the real life situation? So far, so good. The results are good. Everything is ongoing. This is our first experiment. Few photos of our first experiment in the Philippines. The plants look good. Look here, look healthy. The feeling looks okay. Here is Paolo. He's the lead of this project, or this part of the project. He also is good photographer, so we are making a record of that. Also, Jenny Lu have been instrumental in this this part, also doing the transformation and so on. I will change a little bit topic, not topic, but disease. And I have one slide. This is blast. This is a different pathogen. It's called Magnaporte oryzae. It's a fungal pathogen from leaves and panicles, and have a similar interaction that XOO, but is also a very big problem. What I want to say here is that there's many genes, resistant genes, that are used to control this fungus. And most of the genes are NBSLRR, basically cytoplasmic receptors. And what I want to say actually is that we are also doing some research on resistance based on starvation mechanism. We are targeting some of these sugar transporters, not sweet as the XO, but other type of sugar transporter that allow to have this, to block this process of importation of glucose and fructose, to block the access of the Haustoria to the nutrient. So I won't talk about this, but probably in the future, I will be happy to discuss with you guys about this. So this is my final slide for this part, and I just want to touch briefly on the actual landscape of genome editing around the world. This is a nice review from Sarah Smith in 2020. I fully recommend you to go through that. It's very nice to read. But this, in summary, is here all the green countries already have or have no regulation for genome edited products as GM, compared to the red countries, which mostly Europe, which have a stable regulation of genome edited product as GMOs. But the most interesting part is all these countries that are in yellow that have now a regulatory process where GMOs are separated from genome edited plants. And this is very good news because these countries are in the front line of research, and probably soon or probably next year there will be some news coming from those areas. So you just keep an eye on that. So to continue with our strategy, the second point that I want to talk with you is about the pathogen itself. Once we have the right gene or the right number of genes that we want, we need to understand the pathogen and try to monitor the pathogen as much as possible in the regions that we work. So we are lucky because for XOO, we have a lot of information. As you remember, this calls bacterial leaf blight. And these are the locations of the genomes that we have been sequencing as a community. There's around 430 genomes across all Asia and across the last 40 years of agriculture in Asia. So we have been capturing this diversity as much as possible. And what we know so far is there are 3 big groups, and there is 15 populations. And 15 populations, each population have its own evolutionary trajectory, its own history, right? Of history. When I refer to history, that's the recombination process, the selection process, the way they accumulate mutations, and the way they interact with the host. So these 15 populations are a key number for us to try to monitor and to understand what is happening with each of these populations. As I mentioned, for these 15 populations that are represented here, we know also the phenotypes, and we know, or we try to understand what are the TAL effector repertoire. So this is telling a lot of how the populations evolve, but how also they have prepared for interacting with rice. And we know, as I mentioned, a lot of these populations. I won't go into each one by one, but each population have its own kingdom and its own way to be controlled, right? For instance, here in South Asia, we have population 4 to 6, and is easily be controlled or can be controlled by one resistant gene that we know. But other populations at 7, 8, probably need 2 genes to be controlled, and so on. So we have this information, and we can modulate the breeding program, and we can modulate the response to the pathogen based on this. We also know that all these populations together globally can be controlled, or can be controlled by a combination of sweet genes. Sweet genes, basically the products that we developed in the previous part, either by sweet 11 or sweet 13, 14 together, or the 3 of us together. And why I talk about population? Because remember, I described how we need to understand the genomic context and the way the populations evolve and the way they carry the genes to develop very simple markers, very simple tools that allow us to fast track this pathogen. So we develop SNP markers that are stable and can be detected high throughput in the leaf. So you just need a piece of leaf in the field, put in a Effector tube, send to the genotyping service, and that's it. So you can have all this information in a couple of days, right? If you remember one of my first slides, to do this in a traditional way take years to try to isolate, understand, sequence, or do many tests. Now we can reduce all this time into a very small fragment of time that allow us to respond faster. If these go to the right person, the right breeder, or the extension service, we can contain the disease in time. And as you see here, we were able to, in 2019, we were able to genotype or profile 1,000 outbreaks across Asia, from Pakistan to Indonesia and in between. So in a couple of weeks, we have a good sense of what is happening in Asia. And definitely we can respond. So what we want is to articulate this and make this a process for each season. So each season we can monitor what is happening with the population in each of these countries. Right now I will jump to BLAST. Remember, BLAST is a fungus. The genome is way bigger, 10 times bigger, is similar to a rice genome actually. And to summarize again, the interaction is very clear. The fungus interact with the plants using this Haustorial that will secrete effector genes, as I mentioned before, and the plant have the resistant protein that detect these genes to trigger a defense response. In the breeding program, we have already several genes we call PI genes. These resistant genes are already in the breeding program. So if we know that these resistant genes detect effector genes, we want to do the same process that we did with XOO, but to BLAST. This time we want to target effector proteins using our markers. And why? Because knowing the frequency of effectors help us to understand how effective is the resistant gene. So for instance, if a effector is 100% present in the population, like here, that means that if I deploy the resistant gene that interact with this effector, it's highly likely that this resistant gene will be effective, right? However, if the population delete the effector or have almost low frequency, that means that your resistant gene will be not so effective or not effective all the time. And we know that this frequency change with time. This is an example of the Philippines. In the old collection, we detect this particular effector, ABRPI-Z-T, at half of the frequency. However, by the new collection several years later, this effector almost disappeared for the same region in the Philippines. So we know that there's a dynamic. So we need to make sure that we monitor that dynamic in time, and of course, more effectively. But of course, Philippines is one part of the puzzle, and what we are looking is the big picture. So to mimic what we did with Santomonas XOO, we went to a similar strategy in which we acquire genomes from all over the world. Our collection from Africa was represented here. We did the collection in 13 different countries in Africa, and also in Asia. But of course, we also use genomes from other publications. And just to give you a summary of the situation, BLAS originated in Southeast Asia, and this was several hundred of thousands years back. And just recently, the last 100 years, probably colonized Africa, probably in the seed system. However, what we find in Africa is a very diverse population. Basically, all the groups that are present in Asia are now in Africa, and these two populations are evolving at different rates and different times, and I will show it. This population was out yesterday, actually, and you can check that here. What I meant here is that when Africa and Asia are having different rates, we measure an index that is called Taijima. And Taijima basically measures the selection process, or the selection process that those populations are being through. And in this case, we know that Africa and Asia have different Taijima values, meaning that they have different selection processes going on. We can observe these values here in a very small fragment of the genome, chromosome 3, I think, of BLAST, in which one of the populations in Africa have different Taijima values to the populations in Asia, and this can be represented or can be represented at gene level. So the gene diversity of these two continents in the same population with the same background pool is different. So that means that something is going on in Africa compared to Asia. This is true for the whole genome, but also true for the effector genes that are our target of interest. And one of the key measurements is the number of effectors. How many effectors that the population have. And when we measure the number of effectors between Asia and Africa, we recognize that there is different number of effectors. And that means that the gain and loss of effectors are also changing and are subject of the interaction. So obviously the hosts in Africa have something to do with this interaction, and the populations are reacting. What we observe here is basically the present absence polymorphisms of all the 178 effectors that we found in the genome. And we need this information. Why? Because, as I mentioned, we need to develop tools or markers that allow us to track some of these effectors. So we develop these markers. At the moment, we are in that process, and we are targeting all the effectors that are being recognized by the genes in the breeding program. So the genes in the breeding program, the resistant genes in the breeding program, have a representation in this set of markers. My point is, whatever we are doing in breeding, we are monitoring in the ground using these markers. So we can advise breeders what to do. If the effector is becoming low frequency, it's better not to use that resistant gene, or it's better to use the resistant gene, and so on. So we're in this process at the moment. This is the final part of the presentation, and I want to talk a little bit on the decision making. So, as I mentioned before, we need a platform to integrate whatever happened with the host on the resistant part, on the breeding part, and whatever happened in the pathology part and the environmental part. So we need a decision platform that allows us to give advice on what resistant gene to use or what variety to deploy the next season. So we create this tool that is called PathoTracer, and is a very simple and very friendly platform that allows decision making on diseases. Again, for a breeder perspective, breeders need to know what resistant gene they need to advance, right? Imagine that a bad decision costs money a lot. So if you advance the wrong resistant gene, then there's a lot of money and resources spent that. And then they need to know this on time. And the second part is, for extension service, you need to know what variety will be resistant or existing variety will be resistant in that location. So we pretend to develop this tool to, again, democratize this information and try to everybody see the same, and so everybody can have similar decisions. And as a visualization tool, it's very simple. You just at least this is bacterial blight. It's named for XO populations. And we try to represent the dominant population in each region. So if you go here, if you click in one of these or provinces or regions, you will have information like the dominant population, what is the collector, what is the resistant gene that is needed, or what is the variety that is out there that can stop this pathogen or have the resistant gene profile that stop this pathogen. You can see here, for instance, in the Philippines, if we click in one of the provinces, we have the profile of this province, but also we have the rice accession that can be used in the breeding process or directly in the field to be planted in the regions with other characteristics as well. So basically, this tool is a platform that integrates resistant and pathogen and environment in one single platform that is simple enough that everybody can have an idea what is happening. And of course, there is a lot to do still, but we are working on that to integrate more diseases, not only bacterial blight and blast, but we expect to have virus diseases and other important diseases in other regions. This is the same platform, and this is a visualization from BLAST. Of course, each pathogen will have its own particular set of visualization. In here are, remember, the effectors. So if we click in this location in the Philippines where IRRI is located, we know that the frequency of these effector called ABR-PIK and ABR-PI9 are almost 100%. That means that if you use any of the genes, the resistant genes that recognize that effector, you are safe deploying that variety. Then I want to finish with this message. First of all, I mean, probably this is the key message here, that no resistant gene will solve everything. If you want strategies that are long-lasting and are effective, you need to understand the host, the pathogen, and the environment together, and try to make a decision based on that information. The second part, of course, if we want to elevate the resistance and the durability, we need to take in account these elements for the purpose of our crops. Right now, some of the resistant genes that are out there are not long living. So after 3 or 5 years, the resistance is broken. So we need to really increase that durability substantially. I have shown you a little bit some examples of new next generation breeding techniques that we are using that are accessible to everyone, and these are targeting different processes as the sugar acquisition, for instance, and we believe those processes are important to increase the level of resistance of some plants. So we are planning to go with more emphasis in the future about those processes. And one last thing is that coordination is a very important activity, right? I mentioned that pathogens move and normally are regional problems. So if only one country or one breeding program or one national program take decision, that doesn't matter. The important thing is that everyone or the region itself take the similar decision. So that's the only way to contain pathogens in the long term, acting together, basically. And with that, I think I finished. I just want to thank many people that somehow contribute with the slides or information in this presentation and are represented there. And our funding sources are not there. I don't know why. but yeah, is German government agencies, Gates Foundation, USAID, BBSRC, and we have also money supporting the RISE CRP, which is a 1CG initiative. And thank you very much for your time.
Thanks, Ricardo. There are a few questions we pasted in the chat box. Do you have you seen in chat?
Wait, I will.
Okay, no problem.
I will stop sharing, right?
Yes, yes.
Chat box.
It's just the green button, beside the green button.
Normally, I have a chat box here. Okay. Okay, I will take one by one. The first one is about Neckblast and how that can be introgressed in popular varieties, I would say. I mean, a lot of people are working on Neckblast. For all of you, Neckblast is caused by the similar pathogen as rice leaf blast, so it's Magnaporte oryzae. How that pathogen infected the panicle is not known, and what resistant genes are tissue specific is not known. But definitely is coming very, it's coming a lot in the rice sector now. Regarding the resistant to those, there is a lot of work going on in China, a lot in India as well. We have been screening for Neckblast using the 3K panel, the 3000 rice genome. And so far we identified 12 accessions that looks promising. And we are actually now in the crossing part, on the breeding part, to try to develop isogenic lines for that. For instance, we identified one lossy using GWAS in the chromosome 12. We don't know exactly what gene is responsible and how efficient this will be in the field. It's very early to say anything about that, but I think there is some concern and there's some effort about this part of the disease. Can I continue with the rest? Or
Yes, yes.
Okay. Yeah, if the SNP or the insertion is enough to block the activation of the sweet gene by the TAL effector. In here is very, what we know about TAL effectors is that they are not static. They can change and they are a little bit dynamic. So if we introduce one single substitution, maybe the protein is dynamic enough to still bind to the promoter or to the region. This is what we know. So that's why we are selecting small deletions or insertions that are probably 2 or 3 nucleotides in size. So and also the position in the sequence is important. If you block the middle of the recognition sequence, then you have more chance that the binding is blocked. That's what we know. Experimentally, is very difficult to test, but some people are working on that. So yes, I mean the size of the SNP or the mutation is a little bit important. What you don't want is a big mutation or big deletion that change the structure of the promoter itself. That's something that you also don't want. Okay, next question. Neckblast, again, any gene shows similar neck and leaf blast identity. Yes, there's reports for some of the genes that work for Neckblast, for leaf blast, and work for Neckblast as well. Unfortunately, when those reports are going to the field, looks like the situation is different. So at this stage, we cannot say that particular gene that is good for leaf blast is working on Neckblast. Very early to say. What are the limitations of ABR genes? I mean, for a detection process, the limitation on work on ABR genes is that they evolve fast. That's meaning that they change fast. So definitely you need to adapt your methodology to that. Biologically, if that's the question, yeah, I don't know. I mean, the gain and loss, the evolution of these genes is very fast. So as soon as they are selected, they can be in the population, but once they are not selected, they accumulate mutations very fast or can be deleted very fast. That's the only thing. Any novel QTL been identified for rice grain quality in the Philippines varieties? I'm not expert on grain quality. I know that the IRRI, I mean, I know the scientist that is working on that, Nese, and there's a lot of work in progress, in particular in the developing good markers, phenotypic and genomic and markers for selection of grain quality. There's definitely some big progress in that area. But I cannot mention that. I mean, I have no idea. It is possible to do pathogen sequencing at IRRI. Yes, we can do. We are doing nanopore because, for instance, for bacteria XOO, tal effectors are full of repeats. So we need long reads to try to assemble the genome itself. Right? If you use Illumina or short reads, you cannot assemble the genome, full genome of the bacteria. So definitely you need or either PacBio or nanopore. Now we are using nanopore. It's possible. That doesn't mean that it's cheaper. Probably it's better to go to a provider that is cheaper than the regular. Okay. Yes, sure.
Yes, you can answer most of them. If you like to continue, you can continue for 2-3 minutes.
Yeah, there's a couple of. How about time? Are we okay in time? Or
yes, for time we are okay.
Okay. So the question is if the ABR genes can work in any other crops. What I can say about that is so the secretion of effector proteins by pathogens is a common process now. So we know now that many of the pathogens that have stable interactions with the plants, crops in particular, develop effectors and evolve effectors to adapt to the host. Whether those effectors can move from one pathogen to another is not well known, but definitely several processes are key. So my point is that to suppress the immunity, for instance, of the plant, different pathogens evolve similar mechanisms. So it's a co-evolution process. That's very interesting. So definitely there's cases in which effectors from one pathogen can be used in a different pathogen to facilitate infection. Yes, that's possible. Eloisa Cortez, how do you cope with? Yeah, how do you cope with fast evolving populations like blast? Yeah, that's the whole point of the presentation, is just being as fast as the pathogen. How we do that is very difficult, but one key part is to being able to detect the pathogen early on. and to have the tools to do that, and the resistant genes in the background. So you need plan A, plan B, plan C, different resistant genes that you can deploy in your varieties in different times, and being able to monitor what is the level of those effector genes in the population. At least for BLAST, that will be my approach. Okay, one is about the identity of the SNPs. So it should be good. It's a comment, like it would be important to make sure that the BLB have an identity test for the SNPs. Yes, definitely. Just to put you in a little bit on the background, what we did is to develop those SNPs is basically go to the 400 genomes that we have and develop SNP markers that are unique for BLB. How we do that? Yeah, it's a very big process in which we have to understand what are the regions in the genome that have recombination process. So you don't want those regions. What are the regions in the genome that are being targeted by selection? So you want those regions. On top of that, you need very conserved genes that are only in bacterial blight, and they're stable across different genomes. So yeah, it's a process of selection until we got those SNPs. Yes, you're right. I mean, cross amplification or cross detection is definitely possible. But we have SNPs in the background that tell us what pathogen may be, if it is a negative or a positive control. Yeah. I think the last question, oh yeah.
Yes.
Yeah. How long does the screening takes? Yeah, I mean, for the screening and for BLAST, yeah, it's a continuous process. Could take a couple of months until you grow the plants and you screen with a representative collection of pathogen and the right controls, and yeah, a couple of months probably to have a good idea of what is in there. Yeah.
Okay. I think it's okay. You have answered many questions, and we collected, and most of questions are interrelated. Like you have answered that IRRI did the nanopore sequencing. Now the followed question is, who is the contact person and address? So it should be available in IRRI website, and that's all.
Yes, I can place there the patho tracer web page. So there's the contact.
Okay.
The contact person there.
Okay, no problem. So now we are moved to the interview round. Shoma, would you like to interview Dr.
I'd be honored to. Okay,
Yes.
so we are now moving ahead to the interview section of today's webinar. Today is our 24th webinar of the series, and we are honored to have Dr. Ricardo Oliva with us today. Hello, and it was a beautiful, very informative and very well explained presentation. We got many appreciation in our YouTube chat, and many people have said very kind words about your talk. So for all of them and all of the people who will also watch later, we want to know more about you as a plant scientist. So you have quite a successful career, and we would love to know how you built your career. And could you please tell us about your journey so far as a plant scientist?
Yeah. Okay, I guess always is. I'm not saying that it's luck, but yeah, you are not thinking about that often about how to build your career. I don't know, people do, but in my case, I am not such a planner. But two things that I take seriously and I recommend everyone is just to get a good supervisor. a good mentor. That's such a help, you know. And there's some books about how to select a mentor, right? Because some people, some supervisors or mentors want you to work hard, right? There's one way, but the others want you to grow. So I definitely select the ones that you think or they want people to grow. That's the key thing. I mean, in my case, I have such good luck with mentors. I didn't do my homework, never investigate any of them, but just by luck, I got in the hands of people that are just such nice human beings, and that growing thing was just probably what kind of helped me a lot. That's one. And the second one, the second kind of not rule, but the second one, even though it sounds a little bit cheesy, maybe it's just you need to do what you want, right? The thing that you really like, because so many of us, we know what we want, but we are or too scared or too, you know, so people know what they want, but they are a little bit scared of taking that decision. Just go ahead. That's the only thing that I can say. So the mentor is definitely one, and the other thing is just the motivation.
Lovely. You have so many fellowships and awards. What is the driving force for your dynamic nature?
You know, I mean, I don't have so many fellowships and awards first, but the ones that I have are a little bit diverse. I would say I see the diversity there, and it's because I try to interact with people that also like questions, you know, people that are motivated. So, and then you can find that in several areas, right? For instance, we are collaborating with people that is doing mathematics, right, and they are doing pathogen things on mathematics, you know, and that people are super motivated, and I like to have that diversity. People doing biotech, we also want to interact with them, and we develop grants and proposals to that. So it's a little bit of looking for people that also have their own motivations, but that the question is diverse enough that get you're interested. Right. So I would say diversity is the key factor for me, at least.
Collaboration.
Yeah, collaboration in different topics. Yeah.
Right. You have published your research work in high-impact journals, and you are also the reviewer of many renowned journals. Could you please give some advice to advice on the factors that we should keep in mind while approaching or submitting manuscripts in such journals?
Okay, so rules there for me: open access. Right, that would be like. The publishing industry has been a little bit, for me, a little bit corrupt in the essence of publications, in which it's not about your work, but about how much you can pay to get published, right? So there's a level of injustice there. So I think that, but in general, open access. The concept of open access we need to follow as a community, and then the next generation we are the next generation, we need to push for that. Then for publishing, I always follow these. There's many presentations about that is called books publish or perish, and I recommend one presentation from Sofiane Camoun from the Sainsbury Lab. On how to publish, you know, step by step. It's really interesting how this can be so easy from his perspective. So he's a very good communicator, and I think that this publication or this video is in the YouTube is very good for any student, any professor, anybody that wants to publish. You know, it's just fantastic, and I always recommend that. And yeah, the publication is always like how do you say this? The publication is always, to publish in good journal is just a motivation, right? That's, you need to really believe what you have and communicate that to the editors, to the reviewers, and try to disseminate this beforehand. I always use preprints. It's very effective now, the preprints. Any platform that is out there, so you can just put that information out there, and then usually the comments that you get are very good, and that will help you to move to the official journal.
All right. So what is your point of view on the food security and nutritional security?
Food security. Oh my gosh, you are taking big questions.
Very important ones, actually.
Yeah. Okay, so that's also a motivation, right? Food security is on the line, I would say. Although I heard some people saying, we are throwing our food, and we just need to distribute food around, and that will solve the hunger and
minimize wastage.
Yeah, which is good. We should definitely do that. However, we are not quite there. I mean, the supply is so vulnerable that we can see right now in COVID situation, right? That just small breaks in the chain will create a big problem in the value chain, and at the end of the day, in the consumer and the producer and farmers and so on. So I think we are not quite there, and the key thing is investing in agriculture, right? So from 2008 to 2012, or a little bit early, there's a big investment, and since 2015 and so on, there's reduced. That's my perception. I haven't seen data, but that's what the people complain, that the investment in agriculture is being a little bit neglected. I think from the government side, the investing in agriculture will be important.
Right.
And yeah, you said nutrition, right?
Yes, nutrition.
Yeah, that's another big thing. Coming from rice, many of these crops are carbohydrates, right? It's source of sugars, basically. And I think this can be a complementary food. Definitely, we need to diversify the diet. That will be key. There's a lot of effort to biofortify our crops. Cassava, rice, wheat, and potato, and so on. They are this is valid and is legitimate, and we definitely support. But in the long run, the food supply and the diet should be more diverse, and so to get nutrients from a diverse set of crops. I think that's the key.
All right.
The long run.
So, according to you, which sectors of plant science research is likely to prosper in the near future?
Okay, probably I am biased a little bit, but I think microbiome and phytobiome areas are key in the next future. Understanding how the plant, the environment, the soil interact together. This concept of food webs, right? So how the carbon cycle, the animal affect the plant. I think this type of research will be key if we want to move to environments that are a little bit tricky in the future. Because the rest, adaptation to stress and so on, definitely we are good on that as a community of scientists, agriculture. We are going a good direction, but in the future we need to add that extra level of complexity, which is the microbial part of, particularly in the soil or anything that have to do with the plant. That's one. Of course, the second, I would say, again, I'm super biased. Probably biotech continue to be one of the important key to develop. And I'm not saying about technically. I'm saying about, yes, technically, yes, but the challenge is also as a communication, how we become better communicators about the benefits of science in the biotech, right? We can see now a lot of pushbacks from sectors that do not accept GMO, for instance, or do not accept genome editing, even though there's building evidence of the economic value for farmers, not even for companies, or that narrative is just going south, I would say. So the technology is there, and there's a lot of room to do in the technology, but also on the communication side, from our side, right?
Right, so true, yes. All right.
And the last part, that I also like, and I think how we grow plants in environments that are not used to that, urban agriculture, for instance. I think that that adaptation should be possible in the future and should be necessary, actually, to kind of take out much of the burden that is now the forest and deforestation and so on. So I need to bring the agriculture to the cities to kind of help the life that's also needed. So a lot of research needs to be done before.
Yes, in small places, more food can grow. Yeah. So, all right. So with the final question, and we can conclude our very, very lovely and wonderful session with you. So the final question is that what would your words of advice be to the new or the younger research fellows out there?
Advice to young scientists. I guess what I see in general is that people start in science and then later quit, and it's just the motivation. How to maintain your motivation, that's very important, because the pathway is not easy.
So true.
A lot of challenges, a lot of issues, not different to other areas, but it's a lot of challenges, definitely. So the motivation is key to keep you going. So definitely you need to select things. If you want to be here in science, you need to select things that you like. That's kind of the key aspect. Otherwise, just doesn't make sense.
All right. It was wonderful to hear from you, and I'm sure all our viewers had a wonderful time. And okay, so going back to Shubha now.
Thank you, Shoma. Thank you, Dr. Ricardo, for your time. And you have mentioned so much informative. There are so much information and well cited. If anybody wanted to know more, your every slide have the reference, and anybody can go and learn more on that. But what I like the most, you mentioned that the pathogen control of a pathogen is not a national job. It's a international and multinational jobs, because if you try to avoid a pathogen in your field, it may goes to another field and your neighboring field, and when its effect of your any chemicals or anything is gone, then they also came back and infect your field. So it's very difficult if you collaborative or community way, you try to avoid the pathogen. It's more better, more easy to handle than a farmer itself repetitively spray. It ultimately increase his or her input cost, but it not so effective. So yes, I also believe that because I'm also work on a insect resistant plants, and it's true for us also. If you improve your field by any other means, it have to be improved your locality. Otherwise, they also keep an alternative host, and they come when the suitable situations arise. In rice, there are these are the more prominent issues, because most half of the people in the world are fed on rice. So rice pathogens are always highlighted, and many people are try to try to build something against resistant on that. And what I like today, the talk, it is a full application of modern techniques. You use genomics, you use the CRISPR, and it not only the showing the path in the future, also it's help to know the better relationship. Because if you not know the relationship fast, then how you can mutate a particular genes and that. So your point of view, it's not, you never mentioned the CRISPR is a technique or that. You took at a technique what should be. CRISPR is just a technique, and you focus on the aim of your research and the outlook to the science community, not the CRISPR or not the genome editing. So it's a nice informations, and very it's a learning process to us also, that you have a plan to build a resistant against it, maintain the yield, maintain the crop health, and you use whatever the modern cutting edge technique are available nowadays. You used, and I think you apply it in such a wonderful way that it brings, as Soma says, is a good paper. Good paper means it's a good work. Not always the good paper. Sometimes the good work not published in a high impact paper, but your all work is a very good, and it's connect the dot as you always focus on the relationships, microbes and plants and environment. So it's a nice throughput for me. It's the take home message that you can do in that way. You can use these available techniques, sequencing to CRISPR, and you can build something which is commendable and neat to the society. Thanks, Ricardo, for coming. I'm hope in future we also I expect you once again when you do your work more little bit. Then once again we should invite you, and let's see what improvement you have done and what we learn from your research, and we can correlate once again. Jacinta, is the bye? Can you see the bye to the speaker? Soma, is it time to?
It was lovely. Your presentation was amazing, and I really thoroughly enjoyed it. Thank you.
Okay, Dr. Ricardo, I sent you a few questions are left there, but it's not exactly related, but maybe it give you some direction or something. And thanks for coming.
Yeah, we will send you the statistics and updates about today's webinar.
Yes.
Okay,
yes.
That's cool. Thank you. Just before you close everything, I just want to thank you guys again. Subo, Jacinta, Liberty, and Soma, because you really rock. You are rocking.
Thank you so much.
I am so amazed of what you are accomplishing. So please keep going with this. You're my hero now.
It means so much coming from you.
Thank you. Yes.
I'm sure we would love to. I mean, we would meet sometime in the future. Yeah, looking forward to that.
We are very happy that you guys also come and to join this platform. It's a little bit new, but our intention is so clear to promote plant scientists, and everything is free for all and everybody, and it's remain free. Yes. So it's
what he's trying to say is that thanks for trusting us. We are new, and new thing, and what it is. So That's a lot of trust from your end. So we are honored.
Okay.
Thanks.
Right.
Okay. Thank you then.
So, okay.
Thanks.
Okay.
See you
Bye-bye.
again.
Bye-bye.
Bye.
Thanks, Jacinta. Thanks, Soma.
Thanks to
for-
Liberty.
Bye-bye.
Yes, thanks to Liberty. Now we have. Can we spare 4 to 5 minutes on our upcoming webinar? Can you share your screen and a little bit update them?
Yeah, sure. Why not? That would be
And
great.
thanks all the participants and the listener. And you may got the feedback link in your email, because it's now mandatory to subscribe before the start the webinar. Webinar registration is very important and mandatory. We have already sent all the registered participants. If you not get it by any chance, but you lost it, you have to email us with today's password. If you're and the response copy that you as a proof that you have registered in our webinar. Many registration processes are ongoing. Shoma will introduce and guide some of it. Okay, Shoma.
All right. So I hope you can see my screen. So this is our website. This is the notice section. Okay, so here you can see all the upcoming live webinars have been enlisted here with the details about the topic and the speaker and the date and the time and everything. So today's session was From Breeding to Genome Editing, Long-Term Strategies to Contain Plant Diseases by Dr. Ricardo Oliva from IRRI, Philippines. Our next webinar is on 4th December. The time is 2 p.m. in the afternoon, Indian Standard Time. Please note the time. You can also check what time it is at your place from the world clock that is provided here. You can click on it and it will show you the time at your place. This is webinar time. All right, so the topic for the webinar on 4th December is translational genomics for achieving higher genetic gains in groundnut. So this is our first webinar on groundnut research. This is by Dr. Manish K. Pandey. He is from ICRISAT, India. Okay, so our next webinar after 4th December is on 19th December. The topic is Agriculture 4.0. It's a future of agriculture. It's by Dr. Dinesh Kumar Singh. He's the group leader at TCS Innovation Lab, Mumbai, India. All right. So the timing is 11 a.m. on 19th December. After that, we have another webinar on 31st December. Yes, the last day of this year, 31st December. And the topic is a journey of rice research from salinity to fluoride stress response. This webinar will be presented by Dr. Arjudeep Roy Chaudhary from St. Xavier's College, Kolkata, India. The timing is 11 a.m. It's on 31st December, right? So after December, we have more webinars in January, that is the new year, 2021. Our first webinar on in the new year, the details are coming up. You will find the details over here shortly. And after that, it's again on 15th January. So this webinar on 15th January is on production of androgenic haploids and double haploids in banana. So this is a first research presentation on banana in Bioengine. It's by Dr. Ramajayam Devarajan from the ICAR National Research Center for Banana, India. The timing is 2 p.m. in the afternoon. All right. So after that, we again have one webinar on 23rd January. It's on the topic Enhancing Yield in Rice, Employing a Systematic Marker-Assisted Gene Pyramiding by Dr. Lakshmi Narayana R. Vamireddy. He is from the Acharya N. G. Ranga Agricultural University, India. webinar timing is 11 a.m. on 23rd January. All right, so after that, our next webinar, after 24th January, we again have one webinar on 1st February 2021. The topic is span genomes in plants beyond a single reference genome. So it's a very futuristic kind of approach, a perspective to plant research by Dr. Shujan Mamidi. He is from Hudson Alpha Institute of Biotechnology, USA. The timing is 11 a.m. on 1st February. All right. So after that, we again have one webinar on 15th February. This is, please note the time. The timing is at 3 p.m. It's later in the afternoon by Dr. Shiv Kumar Agarwal. He is from Icada, Morocco. The topic is widening the genetic base of legumes through pre-breeding. All right. So as of now, these are the webinars that are available for registration. You can register here. And when I'm talking about registration, I need to stress on it that you need to register for many reasons. One of them being that it helps the speaker to get an idea about his audience and how he should build his webinar around which type of audience and the jargon that he needs to use for the people to understand. So please, when you fill out the forms, you need to put in all the correct details about your name and what are you doing currently. Whatever are there in the form. Your college name or your institute name, everything. And most importantly, please put in your email address correctly. If you put in an incorrect email address, you will not be able to receive a mail from our end which will contain the feedback link, because we are sending the feedback link via mail to all the registered participants. You have been watching that we are not sending the feedback link, the certificate application link in the YouTube chat. You will only receive it via email, and you can only do that if you send in your correct email address in the registration form. All right, so I'm scrolling down to the bottom part of the website where you will find that we have our upcoming live discussions posted here. So this is a completely free and open to all discussion section. You can all just log in to your YouTube and you can watch it at the specific time. Very relevant topics are being discussed here, which are very relevant and very important for new researchers who are looking for help, or you're looking for some just to see if what others are doing and how I should go about my research and my presentation and manuscript writing and my when I click my pictures, what should I do and what don't do. So very essential things, and there have been 2 such discussions in the past. They were on 7th and 17th October and 7th November. The topics that we are already discussed are, credit a rapidly adapted system by international journals to determine the role of the authors and their area of contributions in a manuscript. So if you have read research papers, you would know that there is a proper way that manuscript that a paper acknowledges or writes or puts it in their paper that who were the people who did what in that paper. So that is the credit system, like who did the writing, who did the thinking, everything. So that's the way to act the person who were involved in the paper. So that's why it is like that, and how you should do it. That discussion was on that. So you can view that. The link is also given here. You can just click on the link and you will watch the video. So there was another discussion that was on a very important one, actually. There was on figure preparation. So everybody who is in research, especially in science, they have to have good pictures to present in their manuscript, because they are the soul of the manuscript. the pictures. People always have, you just flip across any journal, you watch the pictures. So pictures are very essential, and Shuvo has very nicely demonstrated and illustrated how you can work properly to prepare such good pictures which are correctly presented. There are many aspects, there are many factors that make or break a paper. One of them is correct presentation of your pictures, of your figures. All right, so you can watch this on our YouTube channel. You just click on this link and the video will start. All right, so most importantly, our next discussion session is on 12th December. And the topic is, tentatively, the topic is around international scholarships for masters and PhD scholars, master students and PhD scholars. So international scholarships, you're all interested in that, especially the master degree holders who are searching for, how to get into PhD. So I hope it would be a very nice learning opportunity for you. Please come into this website and just see when how is it going about. All the news is here about the discussion section. So 12 December around 2 p.m. you will be able to see the discussion session. Next one. All right. We are also accepting requests from researchers who want to present their research on our platform. So here is the link where you can click, and you will find there's a form, and you can fill out the form and put in your details that I want to present this type of topic, and I am this background. So we can maybe talk to you later about what you want to present, mail you, approach you. All right, so, and this here you can see that all of our webinars that were in the past are listed here. This is not just any list. This is the place where you have to come to get your certificate. So the latest one is at the bottom. So if today was our 24th webinar. So here you will find that up to webinar 23, all the certificate have been provided here. So if you want to take your certificate, which you have applied, which you are applying, which you will apply for our today's webinar, you come here to this section, and after 2 days, we need 2 days time for all the process, making all the certificates and all. And you will find that webinar 23 and everything detail is here. You can click on the feedback forms are here. The feedback forms that we received, they have been sorted out, and the names of the candidates who won the certificate are enlisted here. So you just search from the Excel sheet, you just search your name and your file number, and then go to the certificate link here and just find your file number and download your certificate. Also, if there are people with the same name, then you search by further, on the side, if the name of the institute and other details are there, so you can tally that, okay, this is not me, this one is me. So if the person has to, if there are 2 same names, then so that's another point that I want to say is that's why we need you to put in all your correct details. This is another aspect of it. That's how you differentiate between 2 people of the same name. All right, so we are also accepting testimonials. So if you like what we do at Bioengine, and if you want to say something about it, so you can submit your testimonials. Yeah, we would be happy to hear from you. All right, Shivu?
Yes, Suma.
Is
Thank
there
you.
anything else that needed to be said?
No, it's okay. Thanks.
All right.
Now we are going to end this meeting, end this webinar officially. And when I stop the webinar, the live chat will close, but video will remain, and it will be remain. But after a few days, we upload also the corrected video, only the talks and the interview session, and cut off the previous and post webinar discussions related to our website and all of. So thanks everyone. Let's see in our next webinar. And there I am planning to discussion on 12. When it's finalized, I email everyone about the topic and the speaker. Let's see what happened to a couple of days. It should be finalized within 2 days. Thank you. Thanks everybody to joining with us.
4th December, right?
Yes, 4th December 2020.
4th December, 2 p.m.
At 2 p.m. Indian Standard Time.
Yeah, in the afternoon.
Okay.
Please note the time. Yep.
Okay. Thanks. Okay.
All right.
Okay. Bye-bye.
Bye.